Hi, my name is Fowkes. I work at FERA Science Limited, and as part of the PGRO Syngenta web series, I'm going to talk about our project looking at viruses in UK peas. As a bit of background, FERA Science Limited is the National Reference Laboratory for Plant Health in England and Wales, and we do both commercial and statutory testing for plant pests and diseases, along with some other services. This was a 3-year project funded by the HDB, looking to develop a generic method to surveillance. And within it, we were working alongside PGRO to better understand what viruses are present in the UK in peas. And this is because of a review we did in 2017, which identified that the latest publication of viruses present in the UK peas was from the 1960s. So there was a gap in knowledge, and this project was to update the understanding. So prior to the project, we were expecting to find a few viruses. These included P. nation mosaic virus 1 and P. nation mosaic virus 2. These are two different viral species that often find a combination. They have a synergistic relationship. P. nation mosaic virus has is persistently transmitted by aphids, and that means it's got a long acquisition time. Long acquisition times mean that chemical control can be effective for the control of this virus. We also expect to find P. seed-borne mosaic virus. This virus has a short acquisition time, so it's a non-persistent aphid transmission. It's got this short acquisition time, and with this type of transmission, the chemical control can be really challenging. However, the main transmission route for P. seed-borne mosaic virus is via seed. So a better way to control for this virus is the use of clean seed. So to actually do this work, Becky and Shona from PGRO travelled to 60 sites across the UK. 20 sites per year for the 3 years. At each site, they randomly chose a 100-metre squared grid, and at each point, they took a plant, and this resulted in about 120 plants representing that site. And these are sent into the lab at FERA. So when they got into the lab, we sampled through a couple of different methods. The first method was to take what we called a bulk field sample. This was a piece of all 120 leaves, and this we tested by high-throughput sequencing. So the reason we tested the samples by high-throughput sequencing because it allows for a broader range of testing. Your traditional methods of diagnostics, that's ELISA or PCR, ask the question, is this virus present? So for example, you could ask, does this seed have seed or leaf have P. seed-borne mosaic virus? And by these methods, the answer would be yes or no. High-throughput sequencing changes that question instead of what is this virus present to what viruses are present, because it looks at what nucleic acid is present in the sample. So essentially that question then becomes, say, what viruses are present? And what this means is that we can identify viruses that we didn't know to be present in the UK, in peas in the UK, or just completely novel. So we also tested the leaves into these mixed-size bulks. And the reason we tested into these, so 15 bulks of 7 leaves, 15 single leaves, was to give a better confidence intervals when we did the instance testing. So we didn't have to think too much about why. Essentially, we tested these mixed-size bulks for the viruses we identified by high sequencing, and this gave us the instance. So this is a table of the results for the 3 years, looking at the 4 main viruses that we found. So it shows the sites, the virus, and then the instance shown as the average instance with the upper and lower confidence intervals around it. Starting at the top, as expected, we found P. nation mosaic virus 1 and P. nation mosaic virus 2. What is interesting, though, is we found P. nation mosaic virus 2 by itself or at higher instances than P. nation mosaic virus 1. And this is unexpected because these viruses have only ever thought to occur together. So that P nation mosaic virus 2 is by itself is interesting. And it's also not just been found by us, it's being found by other labs, including Germany in their virus surveillance. However, at this point, we don't understand what P nation mosaic virus 2 is doing, what the symptoms causing on the plant, if it's causing yield loss, how it's moving around, and that would require further work. We found PC-borne mosaic virus, but only in years 2 and 3. And as you can see, we're sort of increasing sites and increasing incidence through the years. We don't necessarily think this is because of a problem in PC mosaic virus. What we actually think is that last year, so 2022, was a exceptional year in terms of weather. So it went from a mild winter into warm spring and hot summer. And actually, through every virus we identified in year 3, we find it at a higher incidence as more sites than we did in previous years. And in year 3, we found extra viruses, which I think is due to these conditions. And last of all, we found turnip-yellows virus. So turnip-yellows virus is known to be a problem in oilseed rape. It's been a problem for many years, but this is the first time we've identified it in UK peas. And it was the most common virus across all 3 years and was present at the highest incidences. And talking about the exceptional weather and increase in virus, in year 3, we found it at all 20 sites. So going more into turnip-yellows virus, this virus has a huge host range. It's persistently transmitted by aphids. So again, chemical control is possible. And it has been reported in Australia by through virus surveillance, virus yield testing, and in Germany through virus surveillance. And that's just within peas. Something interesting about this virus is it's known to be symptomless on pea. So this photo on the right here shows a virus that is a plant that's infected with turnip-yellows virus, but is no associated symptoms. And within the specific yield loss work done by Australia, they also looked at symptoms, and there was nothing typical of virus. So no yellowing, mosaicing, stunting symptoms. So additionally to looking what viruses are present, there was a want to try and understand the possible impact of these viruses on the plant, on the yield. And we managed to do this for P nation mosaic virus 1 and turnip-yellows virus. So these are the results of P nation mosaic virus 1 for each of the years. So for this, Becky and Shona went back to 4 or 5 sites each year and collected peas from both the treated and untreated sites, untreated parts of the site. And the way to sort of understand these graphs is the steeper the graph, the higher the yield loss. And what you see for P nation mosaic virus 1 is that there is no difference in yield loss between treatment and treating the site, and there is no difference between the treated and untreated sides of the site. Whereas turnip-yellows virus, what we find is that where the crops have been untreated, you see a high yield loss. So these models, depending on what year you look at, are predicting about 40 to 50% yield loss, where treatment is not used. So where treatment's used, you don't really see as high a yield loss, but it's that potential to the yield loss without chemical control. I say these are all predicted models. The Australian group did a specific yield loss study, and they predict about 45% yield loss for turnip-yellows virus in pea and about 36% in lentil. So again, it's matching to what we're predicting, but in a different agricultural setting. Additionally, within the survey, we found a few other viruses, and that includes P. necrotic yellow dwarf virus. This is the first finding of this virus in the UK. It was found at one site in year 2, and it's persistently transmitted by aphids, so chemical control is possible. What's concerning about this virus, though, is it's got the potential to cause huge yield losses. So this picture on the right shows a healthy pea and a pea that's infected with P. necrotic yellow dwarf virus. It causes these stunting, yellowing symptoms. It was first found in Germany in 2010 and has since been found in Austria, Denmark, and the Netherlands. And there is work going on in Germany where it's really widespread to look at resistant varieties and the effect of Pinocrotic yellow dwarf virus on physical properties of peas. And what they found in that work is that if the plant is infected at seedling stage, you get 100% yield loss because the plant just doesn't grow, it just dies off. So that's why it's a concern of it coming over. Additionally, we'd say we found some extra viruses. We found soybean dwarf virus, and we found this across all 3 years, but it was sporadic findings and very low incidence, so it's not really a concern, but it was the first finding of this virus in the UK. Additionally, we found bean yellow mosaic virus and bean leaf roll virus. Both these viruses are known to be in the UK. Prior to this work, we had sporadic findings of bean yellow mosaic virus, and we only found bean leaf roll virus in year 3. With bean leaf roll, we had a couple of low incidence findings, whereas bean yellow mosaic virus, especially later in the season, we did get some very high incidence findings. Last of all, we found a couple of cytorhabdoviruses. So this is a genus of virus. We found cabbage cytorhabdovirus 1 and Trifolium pratense virus A. These are first findings for the UK and first findings within P. These are relatively new viruses. Cytorhabdovirus cabbage virus 1 has only been found in a Greek cabbage, and Trifolium pratense virus A was reported last year in Trifolium pratense, which is a clover, in Czech Republic. So again, we've not got any idea of what sort of yield loss or symptoms they could cause on peas. So just to conclude, this survey and this work has really been helping to better understand what viruses are present in UK peas and their incidence. Through this work, we've had first reports of actually 5 viruses in the UK. So in UK peas, so that's turnip yellows virus, soybean dwarf virus, and Pinocrotic yellow dwarf virus, and then those 2 cytorhabdoviruses I briefly mentioned. And altogether, this goes towards management in the UK of viruses in UK peas, understanding what and where chemical control could be used. And going into further work, it's looking at turnip yellows virus, for example, so the resistant varieties and where it's coming from. So I mentioned that turnip yellows virus is a big problem in oilseed rape. So there is a question of whether this is the same turnip yellows virus moving between peas and oilseed rape, or whether there's weed intermediates, how turnip yellows virus affects the pea. So in terms of marketability, in terms of nutritional content, etc. And finally, we've shown that we can successfully integrate this technology, high-throughput sequencing, into a surveillance workflow. And next, for that particular point, would be looking at how we could apply it to different crops. This is just a quick thank you to all the hard work that's gone into this survey, into this project, from both the teams at FERA, at Becky and Sean at PGRO, and our colleagues around the world who are also working on pea viruses. And last of all, if you have any questions, feel free to email me at my email address. I've also included the team's email address, virology@fera.co.uk. And lastly, any inquiries on testing, mycology, bacterial, fungal, nematode testing, etc., that you have, if you just email the plant clinic, we can try and help you. But say any of those email addresses, you can get in touch with us. Thank you very much for listening.